Hi students, next we are going to discuss about the important protein chemistry 5-mark question is denaturation, right? Denaturation of proteins. So what do you mean by denaturation? That is definition and principle. What are all the denaturing agents, how it is classified, and what happens after this denaturation of proteins, and finally how it will affect the biological activity, right? Either it will decrease or it will be increased. And finally name the covalent and non-covalent bonds. For this denaturation you should know about the bonds. That is, it is divided into covalent, that is strong bond, non-covalent, very weak bonds. So what are all the examples for covalent bond is, number 1 is peptide bond, number 2 disulfide bond. Example for non-covalent bond is hydrogen bond, hydrostatic bond, van der Waals forces, electrostatic forces, right? This is the basic for the bonds. In denaturation, how the bonds will get affected, which one is intact, what are all the bonds are affected, like that we are going to discuss. So what is the meaning of denaturation? That is alterations in the native structure of protein. What is the native structure? Usually in the structural organization of protein we are having 4 type of structural organization. First organization is primary, second is secondary, third tertiary, fourth one is quaternary structural organization of proteins. In this, native structure will be affected by denaturation. So that means folded protein will become an unfolded protein by some denaturing agents. Second one is, in the structural organization, the primary structure is always intact. Why? Because primary structure is very strong bond. It gives structural strength and stability for the proteins. And another important point is the peptide bond is not hydrolyzed. All the other secondary, tertiary, quaternary will go for hydrolysis and get dismantled, whereas primary is always intact. Third one is secondary, tertiary, quaternary will be dismantled because they are stabilized by hydrogen bond, hydrostatic bond, electrostatic bond, and van der Waals forces. All are weak bonds, right? So what are all the denaturing agents? They have divided into 3 groups. One is physical agents, another one is chemical agents, third one is some physical, mechanical and chemical agents are called denaturing agents. That is, heat, ultraviolet light and X-rays are physical agents. Mechanical shaking, vigorous shaking of the solution or protein will go for denaturation. Chemical, strong acids and alkali, heavy metals like lead and mercury, and some urea solution and salicylate. These are all some miscellaneous. So first one is physically, mechanically, chemically. These 3 are the very, very important agents for denaturation, right? And the next one is, suppose after denaturation, what will happen to that protein? Increased digestibility. That is why after cooking only we will eat most of the food items. They will go for denaturation, then the digestibility will be increased. Second one is usually irreversible. For example, we are preparation of omelette from egg, that is usually irreversible. Third is, it decreases solubility, and finally they will go for loss of biological activity. This is very, very important. After denaturation, the proteins will go for biological activity, loss of biological activity. And already discussed about the bond, covalent bond and non-covalent bond, important classification for your denaturation question, right?